Ever wonder how a tiny anther can release a cloud of pollen? Today, we'll zoom in and watch pollen get made, step by step. In this video, Microsporogenesis making pollen, you'll learn how pollen forms inside the anther. Microsporogenesis comes from two terms. Microspore refers to the young pollen cell, and genesis means formation. A flower is the reproductive part of the plant. The male part is the stamen, made of a filament and an anther. The anther is where pollen is produced. Some anthers have one lobe, and they are called monodithecous. Some have two. A two-lobed anther is called dithecous. That two-lobed layout matters because it sets up multiple pollen-making chambers. If we cut a typical bilobed anther and look inside, we see tube-like chambers called pollen sacs. Usually, each lobe has 2 sacs, so that's 4 pollen sacs total. Now let's zoom in into one pollen sac. Its wall is built in layers. The outer layers help protect the developing cells inside, while one special inner layer plays a huge role in making pollen possible. The outer layers are like a protective jacket, but the star of the show is the tapetum, the innermost layer lining the sac. It supports and feeds the cells that will become pollen. Inside the sac are many cells called sporogenous cells. Not all of them will successfully become pollen, but the ones that do begin as a special cell we can single out. This cell is a microspore mother cell. It starts with a full set of chromosomes because it belongs to the plant's main body stage. Pollen needs half the chromosome number, so the microspore mother cell must divide using meiosis. Meiosis is the type of division that reduces chromosome number by half. After meiosis, one mother cell becomes 4 cells stuck together, called a microspore tetrad. They're held in place by a temporary protective material called callus. Those 4 cells can be arranged in different patterns, like a cluster, a line, or a T-shape. The exact arrangement can vary by plant, but the key idea is the same. 4 new microspores form together. Next, the tapetum helps separate them. It releases an enzyme called callase, which breaks down the callus so the 4 microspores can split apart. Once they separate, we have 4 individual microspores. That moment, forming microspores from the mother cell, is microsporogenesis. These microspores will later mature into full pollen grains. Let's do a quick count. If a pollen sac has 12 microspore mother cells and each one makes 4 microspores, that gives 48 microspores from that one sac. And because a typical bilobed anther has 4 pollen sacs, you'd multiply 48 by 4. That's 192 microspores from a single anther, even with just 12 mother cells per sac. A common misunderstanding is thinking one anther makes only 1 pollen grain. In reality, each microspore mother cell makes 4 microspores, and an anther contains many mother cells across multiple sacs. So far, we've covered the anther's pollen sacs, the tapetum's support role, and how meiosis forms microspore tetrads that separate into individual microspores. Ready to lock it in? Tap the link for study guides and flashcards on this topic, so you can keep learning without missing a beat. Now let's deepen the idea. If the tapetum doesn't function properly, microspores may not get enough support, or tetrads may not separate well, leading to fewer viable pollen grains. When you see an anther diagram, try this checklist. Find the 4 pollen sacs, find the tapetum lining each sac, spot the mother cells inside, and look for tetrads or separated microspores, depending on the stage. Also, let's remember, anthers can vary. A single-lobed anther typically has fewer pollen sacs than a bilobed one. Fewer sacs usually means fewer places to produce pollen. So while anthers may look different on the outside, the process inside follows the same basic pattern. turning one mother cell into multiple microspores. Microsporogenesis gets us to individual microspores. Next, those microspores change and mature into pollen grains that can travel to the female part of a flower and help form seeds. To recap, the anther contains pollen sacs. Inside, microspore mother cells undergo meiosis to form 4 microspores in a tetrad. The tapetum supports development and helps separate the microspores so they can mature into pollen grains.
Now that you've watched the video, let's see how much you remember. Head to the link in the description where you'll find Quizlet content made just for you.